For all of you who don't know me yet, I'm Schneider. I'm working in the feeble plant breeding group of Monika Messmer. And yeah, I want to elaborate on this question on to how to characterize populations. The question arises how to differentiate these, and one aspect you can do for sure is perform some phenotyping, go out on the field. And another aspect that I want to highlight here is perform some genotyping. You can go into this topic of genotyping by looking at this on two levels. The first level is by checking the genotype frequency in your entire population, which means that you have to test quite many genotypes individually. And on the other side, you can go for the allele frequency, which allows you to test these genotypes in a single pool sample. And this allows us to save a lot of time in the preparation of the sample, DNA extraction, and so on. And you can also see details with an infinite depth, so you can see even down to gene level what has changed over the time. And it will definitely simplify, simply reduce the cost massively. I will come back to this later. In general, I want to emphasize this on one case study we performed and created this method. What has been done? Cultivar was crossed with a wild form spring barley. We generated a BC2F3 population, and then these two were sent into a conventional organic farming system for about 20 generations with no artificial selection. Our aim was to identify variations in the genomic composition and basically in the allele frequency, which have occurred after this 20 years of adaptation to these systems in these two populations. How did we do that? We performed some genotyping of pool samples. So we selected some of these generations, two samples for each of these generations with about 300 genotypes each, and performed then a whole genome sequencing of this entire genome with a coverage of 10 times of each base pair. And then a result of this is shown here on the right side. So you can see here nicely this conventional and organic agroecosystem and these generations here popping up in different colors. And you can nicely see that you can differentiate not only the systems nicely apart from each other, but also these generations. So even within the same population, you can differentiate these generations nicely from each other. And one additional thing you can see and kind of monitor is also by the distance of these points here in this principal component plot, how diverse is your population. We haven't done this only in spring barley, also we started with this, but we also performed this in winter wheat and in winter rapeseed and found some kind of same patterns, and the method is valid across different species. And in winter rapeseed, because of the small genome size, the costs were quite low. We performed this for each consecutive year we had a seed available from, and you can see that even from one year to another we can identify variations. So mentioning the costs, we performed kind of a high resolution, even though it's still quite very low, and the prices related to this were per sample given down in here. But maybe for you as a breeder, price is a relevant aspect, so you could even reduce the costs even more to reduce these aspects here. As we did this high resolution, I want to just highlight one aspect where you can go one step further, because it's not only possible to differentiate these samples from each other, but you can annotate this to the genomic region. and, for instance, identify variations coming here from a starting population here on the third chromosome, for example, with a wild form allele frequency, and then see how that changed after these 20 generations, where we, for instance, see here that this organic system has increased in its allele frequency quite drastically, while we have a depletion of this wild allele frequency under conventional systems. And you could go even one step further and saturate this with additional information when you allocate these traits to specific regions, where you can find then some QTLs annotated to these regions, in this case here allocated to yield components. And when you aggregate this across the entire genome, you might also end up with such an your matrix where you can define your population across the time periods and identify how it has changed and which kind of attributes it now facilitates and which are depleted. Yeah, this is the end, and I'm looking forward for these breakout room discussions with you. Thank you.